Hi, my name's Rosie Masui, and I work at the Kachemak Bay National Estuarine Research Reserve in Homer, Alaska, and this is
Jasmine Maurer.
And together we make up the team that works in the Harmful Species Community Monitoring Program. This program has been supporting the South Central region, looking for harmful algal blooms and marine invasive species since 2006. The program is supported by a state wildlife grant, and that's federal dollars that are designated to us by the Department of Sportfish in the state of Alaska. Our partner that is providing match this year is the Smithsonian Institute. So today we're going to provide some information about the two sectors of our program and the regions that we support. To get things started off, I'm going to talk about our Harmful Algal Bloom Community Monitoring Program. For those of you that are not familiar with harmful algal blooms, these are when different species of phytoplankton are increasing in abundance and they are causing some sort of negative impact, either on the marine ecosystem, on marine mammals, seabirds, fishes, or the economy. But the main reason that we're really focused on them is because of the negative impacts they can have on human health. And that's in relationship to causing things like shellfish poisoning. The state of Alaska does not have a statewide monitoring program, so if you were harvesting shellfish for recreation or for subsistence, you do not have any information about whether or not those different species of shellfish are going to be holding any toxins. And that's where our program comes in. So we are not a regulatory agency by any means, and so we partner with different state departments if we do detect any sort of toxins in shellfish. Our HAB program can be split into monitoring phytoplankton and then also monitoring shellfish in case of toxins. We have monitors that we work with all the way from Lower Cook Inlet through Prince William Sound, and we have a great diverse group of community monitors that we really appreciate providing support for this project. They're concerned about harmful algal blooms because of the potential impacts they could have on the economy, on human health, and how they could potentially impact their way of life. Our monitors are doing some protocols to collect the phytoplankton samples that are pretty simple. You can see here I have a phytoplankton net, and at the bottom is a sample bottle. What our monitors are doing is they're lowering this into the water, they're setting it down so it's at 1 meter depth, and then they're porpoising it up and down, just like this, for 3 minutes through the water. At the end, they pull it straight up, and what they end up with is a concentrated sample in this bottle. You'll notice that there's a label on the bottle, and what we have our monitors record are information like, what are the tides doing? What's the weather? What's the water temperature? Because all of this information can help us understand things about bloom dynamics of phytoplankton in general, not just about the harmful algal bloom species that we're concerned about. They then send those samples into us, and we look under the microscope. We're recording all the different species that we see. That's one of the things that makes our program unique. We're not just recording the potential harmful algal bloom species, because a lot of those species of phytoplankton are really important for our ecosystem and can provide really important things like great food sources for filter feeders. After we record that information, we share with folks through a weekly update what we're seeing in our phytoplankton samples. We also have a few community monitors that we're working with that are doing things like doing the microscopy and looking at these samples underneath the slides as well. One of the things that we've learned throughout the years of this program is that just monitoring the phytoplankton only tells us one piece of the story. So we're really not getting the information about whether or not shellfish are becoming toxic from some of these species of plankton until we actually test the shellfish themselves. How we do that is we are primarily looking at shellfish in the greater Kachemak Bay area and a few sites in Lower Cook Inlet. We have a few different mussel cages, just like this one, that are stationed around Kachemak Bay that have mussels that we hang in them at the beginning of the season. Then throughout the season, we grab mussels from here, and we're sending them in to one of two labs. There's a lab in Anchorage that we work with, and that's the Department of Environmental Conservation's lab, and they are able to tell us whether or not our shellfish are toxic or holding toxins above the regulatory limit. And then we also work with a lab in Sitka as well. That's the Seator Sitka Tribes Lab, and they're also able to do the same analysis for us. And then, like I mentioned previously, we are not a regulatory agency. And because of that, it's really not our job to tell folks if something is safe or not. We are more the messenger. So if we get information back from one of those two labs that says the shellfish is toxic, what we then do is we contact the Department of Health and Social Services in Anchorage and our local Division of Public Health here in Homer, and we ask them to work with us to develop a public service announcement that we then help distribute to warn folks that there might be toxic shellfish in their region. The information that we get is really important to have for folks who are trying to make decisions about harvesting wild shellfish in our area, and we really could not do this without our community monitors. If you'd like more information, we'll describe and list some resources at the end of this video, and we will also have some links listed in the description for this video that you're looking at right now. And with that, we're going to pass it over to Jasmine Maurer to introduce and talk to us about our marine invasive species monitoring program.
So Rosie just gave us an introduction to our harmful algal bloom program, and I'm going to talk now about our marine invasive program. At the Research Reserve, we monitor for marine invasives so that we can detect them early, because we know that that is the best way to prevent them from causing harm to our environment, our local plants and animals, or human health. We monitor for two invasive species in the marine environment. The first one are tunicates. Tunicates are an animal that attach at their base to a rock, a boat, or a dock, just like an anemone would. They filter feed. We do this program in partnership with the Smithsonian Environmental Research Center, and we follow their protocols they use throughout the United States. There is a 4x4 PVC plate we attach to a brick and then hang off of a dock on a line. During the summertime, we come back every 4 to 6 weeks and look to see what is growing on that plate. We identify everything that's grown on that plate, and in this way we can monitor for potential marine invasives or invasive tunicate species that might arrive here. Again, early detection is our best way to keep these species from causing harm and establishing themselves in an area. The invasive tunicates can really cause problems by covering a boat and causing a lot of drag, preventing animals from being able to filter feed or access water. They just smother everything by growing so quickly. And so that is the reason why we're taking a look for these invasive tunicate species. The other species we monitor for are European green crab, and we do this with the help of our community monitors throughout Kachemak Bay. Each one of the monitors has 5 of these folding traps they set out at low tide. They're baited with herring, and then they're allowed to soak in the water for 24 hours before coming back. We can then open up the net and identify everything that is in there, all the local crab species or fishes that might get in there. All the native species are released alive right there at the site. I do want to say that so far we have not detected any invasive European green crabs or tunicates in Kachemak Bay. Again, this program has been going on since 2006. And we just really want to thank our community monitors that help us get a wide coverage throughout Kachemak Bay and each of their different sampling locations. If anyone's interested in learning more about tunicates, we do have a guide to some Alaska tunicates available on our website, as well as a local guide for our native crab species that you could look at before going out the next time at low tide. And then of course reach out to Rosie or I with any questions you might have. We'd be happy to answer them.